Immunotherapy is a cancer treatment that uses body's defense mechanisms to eliminate tumors. The human body uses T cells to target affected or abnormal cells. However, cancer cells can block these mechanisms. To resolve this, scientists have genetically modified the patient's own T cells. A disabled virus is inserted into the T cells, which carries genetic instructions to allow T cells to grow artificial receptors. These are called chimeric antigen receptors. This therapeutic approach has been successful in targeting B-cell malignancies, which contain the cell-specific antigen CD19. However, it is not specific to abnormal cells and targets normal cells as well. There remain many limitations to the CAR T-cell therapy. The focus of most engineered T-cell strategies is on identifying and targeting a single tumor-specific antigen with CAR. The drawback to this is that the tumor-specific antigens are rare. Another limitation is that antigens that could be targeted are overexpressed in solid tumors and also expressed in bystander cells in lower levels. In this report, an improved technology has been proposed and tested that will allow to specifically apply CAR T-cell therapy to discriminate tumor cells from bystander cells. The approach began with introducing multiple receptors that detect tumor antigens and tumor-specific antigens, which allow the cells to evaluate the environment and perform more precise tumor killing. Synthetic biologists have contributed by synthetically modifying the Notch receptor to replace its sensing and response domains with recognition and transcriptional domains, thus creating the Syn-Notch receptor, which can then be incorporated into CAR T cells. In this study, a Syn-Notch receptor circuit was designed to activate only when T cells detect two antigens. When antigen in green is detected, it will trigger expression of the CAR receptor, which will in turn activate the antigen in purple. So the cell will only kill a cell when both antigens are detected, and bystander tumor cells will be ignored. Experiments have been performed in vitro and in vivo to test if the Syn-Notch gated CAR circuit does really work in living organisms. The first attempt to test the Syn-Notch receptors was done by engineering combinatorial antigen control over the activation of Jurkat cells. The experiment targeted two model tumor antigens, CD19 and mesothelin. Since the Jurkat cells only express the mesothelin CAR in response to CD19 Syn-Notch stimulation, it was hypothesized that the T cells should not activate in response to mesothelin alone. The results obtained from tests performed on Jurkat cells confirmed the hypothesis, as activation by only the tumor cells which express both CD19 and mesothelin was seen. The graph obtained from an ELISA experiment showed that cytokine production was only apparent when the Jurkat cells were exposed to both antigens. Similarly, the histograms showed the activation of marker CD19 in the Jurkat cells to be expressed only when T cells were exposed to both antigens as well. After the success of the Syn-Notch circuit in Jurkat cells, the same type of Syn-Notch-driven CAR expression circuit was tested in primary T cells. The cells were engineered with a GFP nanobody, Syn-Notch receptor, and CD19. It was hypothesized that these gated primary T cells should only activate and kill cancer cells if both antigens are detected. The results obtained from tests performed on primary T cells confirmed the hypothesis, as activation by tumor cells only which expressed GFP and CD19 was seen. The histogram demonstrated that CAR is only expressed when GFP is present on the surface of the target cells. Similarly, from the flow cytometry plots, it was seen that T cells killed 87% of the dual antigen cells. Engineered T cells with GFP and CD19 were injected into the left side of mice with Dowdy tumor. It was observed that the engineered T cells ignore the bystander cells with the single antigen. But in the tumor, it was seen that T cells became armed and activated. In a highly selective manner, the T cells killed the tumor cells. The results obtained from tests performed on mice again confirmed the hypothesis. Two graphs were produced, with the top one showing tumor volumes for mice treated with SynNotch CAR T cells, and the bottom one with the control T cells. It was seen that the dual antigen tumor was cleared out and the single antigen tumor was left untouched. Similarly, in another set of results, 5 mice were treated individually and showed selective killing for the dual antigen tumor. Through this experimental report, the design of the SynNotch receptors was found to be successful. The combinatorial antigen sensing characteristics enhanced the ability of therapeutic cells to recognize and eliminate tumor cells. Furthermore, using multiple antigens efficiently provides high power to discriminate between normal and tumor tissues. With the design of SynNotch receptors still in progress, they could be programmed beyond dual antigen recognition for enhanced specificity.